Gene expression from genes to proteins. As you should know, our genetic information is stored in our DNA in the nucleus of our cells. Genes are a specific part of DNA strand that code for specific proteins, which determines what we look like and how our body works. DNA is the control of all our protein production. DNA stores the genetic information in the nucleotide sequence. 4 DNA nucleotides must code for 20 amino acids. Amino acids are the monomer of proteins to make a full protein. The 2 nucleic acids you are going to need to know for this process are DNA and RNA. DNA, or deoxyribonucleic acid, has a deoxyribose sugar and is double-stranded. It contains bases adenine, which pairs with thymine, and guanine, which pairs with cytosine. DNA holds the original code for our genes. RNA, on the other hand, is short for ribonucleic acid. It contains a ribose sugar and is single-stranded. It has the same bases as DNA except for one major difference. It contains uracil instead of thymine. So an A is going to pair with U instead of A pairing with a T. RNA holds a copy of codes for genes. Your first keyword is DNA. There are 2 parts to the protein process. In making proteins, the first part is called transcription, when the DNA is used to make RNA, specifically messenger RNA for this situation. The second part is called translation. This is when that RNA is coded for amino acids, which is then going to make a protein when they are all linked together. This is an image of where the process is going to take place. Transcription takes place in the nucleus of the cell, as you can see the DNA coding for the RNA. Translation is going to take place in the cytoplasm of the cell and then move to the ribosome. Your second keyword is AND. Part 1, Transcription. In this part, one of the DNA strands is used as a template for making a complementary RNA strand. An enzyme called RNA polymerase is going to add the nucleotide onto the RNA molecule as it reads the DNA strand. Messenger RNA is made once the process is complete. This is a single-stranded copy of the DNA gene sequence used to make proteins. Here is an example how RNA is synthesized from DNA. On the left side, the DNA nucleotides and the RNA bases are added to the RNA molecule on the right side. Adenine pairs with uracil. Remember, it is not DNA, it is RNA. The T pairs with adenine. Cytosine pairs with guanine. And guanine is going to pair with cytosine. Be sure to note that this is making a separate RNA molecule, not DNA replication. Your third keyword is RNA. Here is an image of transcription taking place. The blue DNA strands are unwound to be separated. The RNA is being made by a single strand of this blue DNA to make a single strand of the brown RNA. The DNA will then wind up again back to normal when the process is finished. Here is an image of the RNA nucleotides being added to the RNA molecule as the DNA nucleotides are being read. Your fourth keyword is R. Take this time to practice on your own by transcribing the DNA sequence into an RNA sequence. Be sure to have your teacher check this when the video is over. Part 2, Translation. Translation takes place in the cytoplasm of the cell and moves to the ribosome. Translation is going to take the RNA that was made during transcription and going to translate it into amino acids to make a protein. Your fifth keyword is used. The genetic code is the key to life. It explains how the RNA nucleotides are going to code for those amino acids. The nucleotides are read in groups of 3. That is a very important thing to remember. Those groups of 3 are called codons, as seen in the picture over here. Each group of 3 letters, or codons, codes for a specific amino acid, which then forms a protein when they are all put together. This is the genetic code wheel. There is also a chart. This is what you will use to find out what amino acid is coded for by the nucleotides or codon sequences. You will always have 3 letters. In this example, we are going to find the amino acid that codes for AGG. You start with the first letter in the middle. You see the 4 big letters. You're going to find the first letter of your sequence in here. So it would be A. Then you're going to look at the second circle for the next letter. Your next letter is G. Now you are going to only look at this corner of the chart, okay, and then you're going to find your G in the next 4 letters. Okay, so our G would be here. And then the next one is G again, so you would look in this little segment, okay, and look out of those 4 letters and you would find G right here. So your amino acid is arginine. Okay, so you start from the middle and then go out. So the middle circle out to the third circle. Your 6th keyword is to, T-O. Take the time to find the amino acids for the given codons above. Use your codon wheel to find the answers and refer to the previous slide for help. tRNA is called transfer RNA and is responsible for transporting the amino acid to the ribosome once it's being coded for. It is like a taxi service. This is an image of the tRNA. See, it is shaped like a T. It brings the amino acid to the ribosome. Here is the amino acid, and it is attached to the tRNA, and it is going to carry it to the ribosome. Your 7th keyword is make. Translation begins when there is a specific start codon. This codon is AUG and codes for methylene. I also remember this by school starting in August. AUG is coded for. The translation process begins and the polypeptide protein chain begins to grow. Since there is a start codon, there has to be a stop codon. And actually there are 3. UAG, UGA, and UAA are the codons that tell the translation process to stop. Once it is stopped, the protein is released into the body to carry out its functions. Practice coding for amino acids and using the start and stop codons by looking over this example. You'll be tested on this when you get your notes checked. Your 8th keyword is proteins.